the GFP story, the origin story of GFP, but maybe from a point of view of how science is done. So let me give you an idea about how I think I learned about science being done when I went through elementary school, from all the stories of the great scientists. The first thing is that scientists are genius. If you're not a genius, do not go into science, because it's just too competitive. It's too hard to do, and you have to have it from birth. I'm obviously telling you a whole bunch of things that are completely wrong, and the GFP story is going to tell you how wrong they are. The other thing we know is scientific stories have to be done in a short amount of time, so we never talk about the failures. So we get the idea that great scientists' work happens instantaneously. The first time it always works. You never have a mistake in science if you're a good genius scientist. The next thing is scientists think different from everyone else in the world. They have this purposeful way of looking at life, which is called the scientific method. They come up with a problem that no one has ever thought of. They realize that there's a way around that problem. They then build a hypothesis, design the perfect experiment, it works the first time, and they get lots of money, or at least that's the fantasy. The next thing about all the things I learned hearing stories about scientists that gave me the wrong impression is that scientists work alone. You know, until Watson and Crick, maybe there was something about people working together, but basically all the great scientists, Darwin, Einstein, everyone worked alone, at least the way the stories went. Now, they may have an assistant, and for some reason the assistant had to always be named Igor, but aside from that, they were isolated and they were loners. And the final thing is that all these great scientists that I heard about, all of them, except for very few people, Marie Curie, George Washington Carver, almost every single one of them was a white man. So from Europe or European descent. And that was what science was. And in fact, if you go down to Google now and say famous scientists, who are the famous scientists? You still get this rather disgusting set of people that are, again, all European or European-derived men, except for a couple people. There's Marie Curie up there. Whoops. Marie Curie, come on. Oh. Anyway, Marie Curie and Rosalind Franklin were on that slide. This is a picture of people that have come through my lab at various times in the last 5 to 7 years. And this gives you, I think, a much better idea about what is happening in the lab. We have people from China, from Peru, from Colombia, from Iceland, from Greece, from Israel, the Gambia, the United States, all over. In the past, we've had people from Korea and from India and everywhere. That's what a modern lab is. And everyone is contributing and everyone is being part of it. So with those erroneous ideas now behind us, let me talk about the GFP story and how I got involved in this. I was working on this animal, the nematode Cenorhabditis elegans, and when the important thing came on, which was not in 1988, but 1989, but I had been working on this animal for 12 years. It's transparent. I said it's transparent in every seminar I ever gave. And after 12 years of saying that, even I believed it. We were also starting to clone genes. And I was really excited about seeing what cells were expressing the genes we had cloned. And so there's a terrific scientific problem. How do you see where a gene is active? Except that had already been answered. It was really easy. You could use antibodies to look where the protein was made, or you could use in situ hybridization to see where the messenger RNA was made, or you could use beta-galactosidase activity that would be a reporter for gene activity, so you could answer the question. But the problem was, in order to do these things, you had to fix the animals. You had to make sure that they were dead, they were fixed, nothing moved around in them, and then you permeabilize them to either get the antibody or the probe or the substrate for the beta-galactosidase in so you could actually see the activity. So what we didn't have is what the subject of this meeting is. We had no dynamic view. We had a snapshot in time, and we really couldn't get an idea about how things were changing over time by looking at this. And it was in 1989 that I heard this wonderful seminar by this man, Osamu Shimomura. Now, let me just break for just a second to say that I have a color code for the names of people on the slides. You may have seen in one of the previous slides that some people had their name in red. Anyone whose name is in red is from my lab. Anyone whose name is in blue is a collaborator. And anyone whose name is in black did something I wish I had done. So the seminar I heard about was about Osamu Shimomura's work to identify the bioluminescent molecule in the jellyfish Achoria victoria. Now, I really urge everyone to go to the nobelprize.org website and read about this man and what he went through in order to be a scientist. It's really a remarkable story. Nonetheless, let me sort of short circuit that to say he was in the United States, he was trying to figure out this, he was grinding up hundreds and thousands of jellyfish to get the biochemical enzymes that were needed to make light, and he failed every single time he did the experiment, over and over and over again. And then one night, the experiment having failed again, he cleans up, wants to go home for dinner, it's already dark outside. He throws everything in the sink that had the overflow for some of the jellyfish tanks, some of the seawater and some other stuff. And he throws the stuff in the sink, turns off the lights, about to leave, looks back at the sink and sees that it's glowing brightly, something he'd never been able to see before. He thinks about it a little bit and reasons that there must be something in the seawater, and that might be calcium, something he never had in a reaction. He goes and he, a couple days later, makes the prep again, adds calcium, and every time he does so, calcium ions, he gets a flash of blue light. And so he names the protein that does this aquarin after the jellyfish, and aquarin plus calcium produces blue light. I want to point out that this idea of throwing things in the sink is a very good bona fide scientific method. People sometimes throw things on the floor or the lab bench, collect them up, and then suddenly they work. I don't really advocate that you do that, but you might try it just, on the side if you're having problems. But he did have another problem, and that is the jellyfish were green and the light given off by this molecule was blue, so he had a second sort of accident. He had the wrong color, and he had added calcium to all his other fractions, and nothing produced green light, so there wasn't another similar protein, but he reasoned maybe there was a converting protein, something that converted the blue light into green, and so he took a handheld UV lamp and went through all his fractions, and sure enough, another fraction had a protein, a different protein, that changed blue light to green. He wrote his paper in 1962 about the purification of aquarin, and in footnote number 3, he sort of mentions sort of on the side, well, there's this other protein, you shine blue light on it, it produces green. I'm going to call it the green protein. We, of course, call that the green fluorescent protein, and it's what, when you add GFP to this whole thing, you get green light. I believe, he is the only person in history to win a Nobel Prize for writing a footnote. So the paper doesn't have to be very long, just something to keep in mind. I'm listening into the seminar, and you remember, I only have a couple of thoughts in my head. One thought is, I work on an animal that's transparent, and I'm interested in knowing when genes are active. And so somebody has just told me there's a protein that all you have to do is shine blue light on it, and you'll see it because you'll see green back. And that's really exciting to me, because I realized this could serve as a lantern, and I got really excited. So what do you do if you're really excited? You get on the telephone and find out if anyone else knows anything about this stuff. And I found this man, Douglas Prasher, who was in the process of getting the cDNA, and we arranged a collaboration. things got screwed up for a couple of years. We lost track of each other, but then we finally got back together when a new graduate student, Gia Skirkin, came into the lab. She had already done work as a master's student on fluorescence, and so she was the perfect person to work on this. And we got back in touch with Douglas. He had the cDNA for us. And this is a page from her lab notebook. 1 month after she started graduate school, she had green fluorescing bacteria. And we knew that all we needed was the protein and it would work. And this is actually a picture she took that first day. There was some problems, and that is she tried to use our microscope. It was a piece of junk. She couldn't see the fluorescence, but she knew where there was another lab where she could actually see the thing. That's why it says she used the scope in the engineering building. But then we couldn't actually do any more experiments because they wouldn't let us on their microscope. Ours was no good. I solved this by getting in touch with all the sales representatives of microscopes in New York City and saying we had just developed a new way of looking at gene expression, but we needed a fluorescence microscope. We were definitely going to buy one. We just didn't know which one it was. And if they could bring a loaner and hopefully leave it for a month or two, we would be able to give it a good workout. And so we did all the experiments on borrowed microscopes. Now, the really interesting thing about this problem is that no one believed it was workable, that GFP could not work on its own. And the reason for that is GFP undergoes a really strange rearrangement. At this position between serine 65, tyrosine 66, and glycine 67, that part of the peptide backbone that's made as it comes off the ribosome undergoes a very interesting change to form a 5-membered ring that allows this molecule to be fluorescent. And no one knew how this rearrangement took place, but everyone was convinced that it was going to take 1, 2, or more converting enzymes. And so people said it cannot possibly work on its own because it needs the converting enzymes. We shouldn't have done the experiment, but we just tried it anyway, and it worked. That said, it is its own converting enzyme. Now, but with that in mind, I have to tell you something else about the experiments we did. I found out there were 3 other groups that tried to do this same experiment. But for them, it didn't work. And of course, they concluded, or could conclude, that it needed a converting enzyme. So what did they do that we didn't, or vice versa? Well, they were careful, and we were sloppy. What do I mean by that? At the time, Douglas's clone was a cDNA clone in phage lambda, and the clone that he had was an EcoR1 fragment that had the coding region for GFP, the green bits, and some other junk on either side. Now, in 1992, when we did these experiments, people had, if you wanted to get lots of copies of DNA, you had to be careful. So the best way of copying DNA was to let an organism do it. So infect a bacterium with the virus and let it replicate. And that's what the other 3 groups did. And they cut this with EcoR1, and they got the green plus the red bits. I don't know why. I was just scared of extra stuff. And so I suggested to Gia that she not do that, but rather do the very sloppy, horrible method at the time of PCR and amplify just the green bit and put that in a vector. The reasoning was we didn't actually care if we made mistakes some of the time because we were going to put this into E. coli, not one bacterium, but millions of bacteria. So even if only 1% of it was the right sequence, if it worked, we should be able to see it. So we were careful in one respect. We didn't have the extra bits, but sloppy in that there was a lot of junk in what we had. Fortunately, though, it worked the first time. Now, after we did that, I want to say one thing. I have no idea how much time I have left because there's no clock running here. So it would be nice if I knew about 10 minutes or 15 minutes before the end. So I can either speed up or slow down. In any case, we went on in 1994 to publish the paper, but not without some problems. One of the problems is science being science. They had to decide whether the paper was worthy of review, and they called me up and said, "We're not sending your paper for review. We're very sorry." And I asked them why, and they said, "Well, frankly, it's because we don't like your title," which I was surprised at because I thought the title was pretty good, Gene Fluorescent Protein, a new marker for gene expression. They said, "You know, everything in the journal Science is new. You can't use that word." Now, I think you can tell how I reacted to that because I suggested then another title, which they accepted to send out to the reviewers. But I think you can tell how I felt by just looking at the title, which was, "The Aquaria Victoria green fluorescent protein needs no exogenously added component to produce a fluorescent product in prokaryotic and eukaryotic cells." This was the entire paper. I could have just appended the figures and that would have been it. Certainly was the abstract. But they sent it off, and then it got accepted. That was nice. But then the editor called me up and said, "You know, do you know your title is a bit long?" And I said, "Oh, is it? Would you like me to make it shorter?" "Oh, if you only could." And so I said, "Well, how about this one? Green fluorescent protein as a marker for green gene expression." "Oh, thank you." So that's what the official title is. I sent in this paper, which I really like because it shows a living C. elegans larva with a nerve cell growing out. And I wanted this to be an advertisement that one would be able to get a dynamic view of life. The art editor called me up, said, "We like your picture a lot, but green is the worst color on the magazine cover. We want to change the color of the picture." This is a very enhanced picture. But it does look horrible if you look at the journal, but I just couldn't get them to do it. And then finally, there were people that were already showing that GFP was working in the organisms they were studying, and I asked people permission to use their work and cite it as unpublished work. Almost all of them said, "Well, you gave it to us before you published. Of course you can," except for this character who wrote in a letter to me that I could cite the work, but only if I made coffee every Saturday for 2 months, prepared a special French dinner for her, and then take out the garbage nightly for a month. And that's my wife. But what Tula did was really the next exceptionally important experiment with GFP. We had taken the control region, the promoter region of a gene, and showed that we could look at gene expression by turning it on. What she did is take the control region, the protein coding region, and then add GFP. So she was the person who made the first protein fusion, and she could watch that protein. And the particular one she did in Drosophila was a protein that went from inside one cell through channels to another cell, and she could actually watch the protein move and be transported, and subsequently do studies of that. So it was very good. She says I've never paid up, but I don't have much more time to talk about this. Obvious advantages of GFP. Its DNA is inherited, so once you put it in an organism, you just keep going. Using it doesn't hurt the animal. Shining blue light is fairly innocuous. UV's not so good, but blue is pretty good. The small size of GFP relative, for example, to beta-galactosidase means that it can diffuse throughout the cell, giving you an outline of the cell. And as I've already pointed out, it allows you to see a living organism. Here are a bunch of different living organisms or living cells with GFP. And if you look in the bottom right-hand corner, you see that Purkinje cell, and you see how amazingly outlined the entire cell is. I keep saying that this is a dynamic view. These are some films that were put on the ASCB website by Rosalind Silverman Gavrila showing nuclear division in Drosophila, an early Drosophila embryo. And I'll start the film off on the left, and you can see this tubulin labeled with GFP allows you to see how beautifully synchronized all the nuclear divisions are as we go through several rounds of division. On the right-hand side, falsely colored, so red means more, blue end of the rainbow less, is GFP with a nuclear localization signal. And if you look at that, you see the nuclei break down. It's all over the embryo. As the nuclei reform, it gets sucked back into them. And you can see the difference in that. So we can actually watch things happen. And then there's a kid's book in the United States called If You Give a Mouse a Cookie. And the main thing about the book is if you give a mouse a cookie, he's then going to ask you for a glass of milk. And if you give him a glass of milk, then he's going to ask you for a napkin. And then he just keeps asking for more. This is a terrific analogy to scientists. You give them green fluorescent protein, and they immediately say, oh, do you have any other colors? And the person who did that, for the most part, did the first one and did many more, also shared the prize with Osama and myself, Roger Chen, who made all these beautiful colors, made fret pairs to be able to make these molecular monitors of all sorts of biological events within here. And of course, many colors have been used for such wonderful things as rainbow, and these wonderful pictures from Josh Zane and Jeff Lichtman. GFP has been used in all sorts of interesting ways to look at fundamental research, to look at applied research. Let me see, fundamental, we look at mutants affecting nerve cell development. Applied here is HIV from one mouse cell being transferred to another, so you can actually see what's happening as a cell transmit the virus to another cell. And then there's been some very strange things that have happened. People have used GFP to try to work as detectors of TNT, to try to see if they could detect landmines. Several groups have done this. The problem is it doesn't always work, and you really don't want a false negative. And so people are still working on it, but I love that they want to try and do this. Something very different from the usual type of experiments that people do. There's a group in Japan that has made fluorescent silk and made clothing out of this. I'm not entirely sure how useful that is because you have to walk around with a UV lamp over your head, but it's nonetheless an interesting thing. And the director Ang Lee had a movie in which he said that this character becomes the Hulk because he's injected with jellyfish DNA that turns green, obviously inserts in a gene where anger turns it on in the skin, and that's why he's green. Let me give some conclusions from what I think this research has really taught me, not those conclusions at the beginning. The first is scientific success comes in many routes. Shimomura was told he actually had to quit high school and work in a paint factory because it was World War II and it was Japan. Actually, that prevented him from dying when the atomic bomb destroyed the city he was in, which was Nagasaki. Roger Chen was brilliant from high school on, won the big Westinghouse Science Talent Search Prize in the United States. And I never show people my grades. So it's very, very different in many different ways. I think most real discoveries come about by accident. The physicist Enrico Fermi has this wonderful line where he says, if you do an experiment and it confirms your hypothesis, you have made a measurement. But if it doesn't confirm your hypothesis, then you've made a discovery. So the discoveries come when you least expect them. And hopefully we'll have our eyes open enough to look at them. I think it's important to be ignorant, stubborn, and just have a willingness to try. Those sort of weekend experiments that you try because you really want to know the answer, but you're embarrassed to tell anyone you're going to do that experiment. If it works, you crow about it on Monday morning. If it doesn't work, just ask people how their weekend went. But you have to try, and the more times you try, the more you get a chance to make those accidental discoveries. Scientific progress is not the result of one person. GFP, the estimate 20 years after the paper was published is that there were 160,000 papers that use fluorescent proteins. And it's really the work of all of those people that made this so important, not the work of the few of us that happen to be honored by this prize. I'll skip this, but well, I'll say a bit. Universities supported me. They didn't care what I did. I like that because I don't like being told what to do. And grant support meant I wasn't working on a specific project. I could use it the way I wanted to, and I never asked people's permission to try the experiment with GFP. The people that really do the work, that really are the innovators in the lab, are the students, undergraduates and graduate students, and the postdocs. Two more things. First, this is a story that starts off with somebody being interested in the fundamental biological problem of bioluminescence and looking at a jellyfish, not one of these model organisms. And I think this says that we should be looking at all of life. And as we see optogenetics, CRISPR-Cas9, all of these have come not from the model organisms, but from really looking more broadly at life. And I think we have to continue that. And the final point I want to make is that fundamental research is essential. We have to have the foundation for any applications. Just as for GFP, for the laser was a terrific basic physics problem that led to many more basic physics discoveries as well as applications. Both are done, both are needed, but you need the grounding of the fundamental research. And I want to leave you with my favorite quote about basic research, which was said in 1969 by Robert Wilson when asked by Congress whether they should build what was called then the Fermi Accelerator in Batavia, Illinois. And they asked him, how will this help the national defense? And he said, it won't. And in fact, he said, it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean, all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much.